Hello, everyone. First, I would like to thank the organizers for the invitation. It's an honor for me to be here and to share my research with all of you. My name is Sabrina Rivero. I am a professor. I'm professor at the University of Seville, where I have just started my own project on Kleefstra syndrome. So today I'm going to present you my recent research. This is an schematic illustration from the long way that goes from the identification of the genetic cause behind a neurodevelopmental disorder, like Kleefstra syndrome, to the generation of a therapy to treat affected patients. The development of a therapy requires a deep understanding of the molecular and cellular basis implicated in the genesis of the disease. And to this end, the generation and characterization of different models is essential. Apart from animal models, the use of induced pluripotent stem cells from patients, as well as model culture cells, has become useful tools for that reason. As a cell biologist, I am interested in understanding the cellular consequence of EHMT reduction in cells. And so our question in mind is how HMT1 reduction affects cell biology. So to address this question, we have a use and sample approach based on the use of siRNAs for EHMT1 downregulation in the RP1 cell line. That is a non-tumor human epithelial cell line from the retina pigment epithelium with a neuroectoderm origin. So using this approach, we have analyzed the possible effect of EHMT1 downregulation in different cell structures and organelles whose defective function has been implicated in different developmental disorders, such as the Golgi apparatus, the cytoskeleton, the centrosome, or the lysosomal system. And we have performed immunofluorescence imaging analysis using different node markers. So using this approach, the first thing we can see is that the siRNAs used efficiently reduce the amount of EHMT1 in cells. We can see that the nuclear staining of EHMT1 almost disappear, and we can see also this reduction at both the RNA and the tubulin, at the protein level. When we analyze the effect of EHMT1 reduction in the structure of the Golgi apparatus, that is a main organelle in the cells with different functions as protein processing and trafficking, theliogenesis or migration, we can see that although the organelle maintains its characteristic perinuclear location, the characteristic crescent shape observed in RP1 cells is lost, and almost all the cells show a collapsed Golgi around the nucleus. So these findings may indicate that EHMT1 has a possible role in the maintenance of the Golgi apparatus structure. In that sense, a similar phenotype has been described in neurons, where this change from a stacked to a collapsed Golgi has been related with synaptic dysfunction. So it will be interesting in the near future to analyze if the possible role of EHMT1 in that morphological change in the Golgi apparatus in cells from different Kleefstra syndrome models. Lysosomes are key organelles for the maintenance of the cellular homeostasis, and in this case, To explore the possible role of HMT1 in lysosome distribution and morphology, we have used an antibody that specifically recognizes LAMP1, that is a lysosome membrane protein. As we can see, cells lacking HMT1 shows a higher intensity of the LAMP1 labeling, as well as an increase in the size of the lysosome. So these results could point to a possible role for HMT1 in the lysosomal system. In that sense, some studies have been identifying that imbalance autophagy cause synaptic deficits in different models of neuromental disorders. So it is possible that HMT1 has also a role in this process. It is also known that alterations in cytoskeleton organization and dynamics is a core dysfunction of different intellectual disability disorders. In fact, it has been described that most mutations affecting synaptic functioning are linked with cytoskeleton regulation, and strong evidence indicates that cytoskeleton dynamics is affected by mutations in different neuromental disorders. However, a little is known about the possible role, the general role of HMT1 in the cytoskeleton organization. So, therefore, we have to investigate the possible role of this epigenetic factor in cytoskeleton organization. We have used we have analyzed different markers by immunofluorescence. When we use an antibody against vimentin, that is a protein of the intermediated filaments that are elements with a structural support function in cells, we don't see any apparent difference between control and HMT1 depleted cells. No difference were also observed when we analyze microtubule using in that case an antibody against the alpha tubulin marker. However, when we analyze when we use an antibody against vinculin, that is a protein that intervenes in focal junctions between cells and the extracellular matrix mediated by actin filaments, we can see an increase in the number and size of these focal junctions, which could indicate that cells are more strongly binding to the extracellular matrix. We can also increase with beta-catenin, that is a protein that mediates the intercellular adhesions. This increase in beta-catenin is clear when we analyze and quantify the protein amount by Western blot analysis. So together, these results demonstrate that in HMT1 downregulation, cells bind more strongly to each other and to the extracellular matrix, which could have important implications for processes as migration. In fact, the essential role of beta-catenin at the synapse has been described, as also has been described the essential role of the Wnt beta-catenin pathways in some developmental disorders. So further investigation will be necessary to understand the possible link between HMT1 and beta-catenin. Next, we have examined the possible role of HMT1 in centrosome functioning. Centrosome is the main microtubule organizing structure in cells, being also implicated in other cellular processes as mitosis, ciliogenesis, and migration. And in this case, we have used an antibody against pericentrin, that is a protein that appears at the central satellites that are these little dots around the centrosome. In that case, we can see that cells lacking HMT1 show we can see a higher number of central satellites and detection. So this result could point to a role of EH21 in centrosome functioning. Taking into account the previous results about the possible role of EH21 in cell addition and in Golgi and centrosome functioning, and knowing that migration process is usually altered in different developmental disorders, we decided to analyze the possible effect of EH21 reduction in the migration process in RPE1 cells. In that case, we can see that after EH21 downregulation, there is cells lose their capacity to migrate in this kind of womb migration assays. Different studies has revealed that the high number of intellectual cognitive genes converge onto few biological processes or modules, as is the case of chromatin factors. In fact, similar cognitive disorders phenotypes are assumed to reflect functional relationships between epigenetic cognitive genes. So in that sense, recent work has described that different epigenetic factors regulate, even they have different functions in cells, a common of cellular genes. So this is the case of EH21, Crib-Vanden protein, NSD1, and CHD2, implicated in Kleefstra, Rubinstein-Tabin, Sotos, and Otis syndrome. So we decided to investigate if the observed phenotypes in RPE1 cells under EHNT depletion were also present in cells with reduced level of CHD8. Between the different phenotypes analyzed, we can see that after the reduction of CHD8 in cells, we can see also an increase in the lysosomal detection and size of the vesicles. And as well, we can see a clear increase in beta-catenin levels after CHD8 downregulation, highlighting the existence of common cellular disruption between these two epigenetic factors. So, in conclusion, we have identified some cellular alterations associated with the reduction of EH21 in cells. However, additional experiments will be necessary to understand the specific molecular mechanism underlying these observations. For that, the use of induced pluripotent stem cells from patients will be a useful option. And we consider that the described phenotypes could open new possibilities to understand the link between epigenetic regulation and underlying cellular mechanisms that leads to Kleefstra syndrome. And this knowledge could be important not only for the treatment of this disorder, but also for other disorders that could share similar common deregulated processes. So to finally, I would like to thank all the people who have made possible this preliminary research, despite our low financial support. And we will continue with the hope of seeing great advances for Kleefstra patients in the future. And thank you very much for your attention.
Thank you, Sabrina. Do we have any questions?
Thank you very much, Sabrina, for the presentation. I have a question. So you have shown that microtubules, that doesn't change, but it is known that the dynamics of the microtubules is very important for maintenance of Golgi apparatus structure. Have you checked it, or do you think it would be some change if you look at the dynamics and have some effect of the lack of EHMT1 in the not only in the organization, but also in the dynamics?
And in that sense, we think that maybe it's a question of that the Golgi apparatus has lost their capacity of microtubule nucleation. So we are going to perform, to respond that question, microtubule repolymerization studies after the treatment with drugs that depolymerize the microtubule. We think that this morphological change in the Golgi is probably related with problems with microtubule organization, cytoskeleton.
So it is known that H3K9 dimethylation is involved in the repression of autophagy-related genes. So I was wondering whether if you have any idea of the changes in lysosomes that you see.
Lysosomes?
Is anyhow related to the fact that the autophagy pathway might be affected in those cells, and whether you have looked at this.
I really don't know. I don't know. I don't know if it is a consequence of is in the origin of the development cell disruption. I have to say that I have been worked in cytoskeleton during some years, but I don't know the specialist in lysosomal system. Maybe you know more than me, I'm sure, in that I don't know in that area. Thank you very much.